Hey Mustangs, in this video we're going to be taking a look at fermentation. Now this is something that happens after glycolysis. So it wouldn't come first, it would come after glycolysis, and it involves the molecule NAD+, and of course its counterpart NADH. Now remember, these are really one and the same molecule, and we'll be talking about that later on. So with fermentation, a lot of our foods, we've actually learned to harness the powers of fermentation in order to make many of your favorite foods. So if you enjoy yogurt, sauerkraut, soy sauce, miso soup, or sourdough bread, those are just a few examples of many foods that we actually use fermentation for. So let's go ahead and take a look at what fermentation is. So at the end of glycolysis, we have made 4 ATP with a net only of, remember, 2, because 2 ATP were used in the beginning, so they cancel out 2 of the 4. So we only net 2 ATP by the end of glycolysis. We've also made, though, 2 NADH, and we've also made 2 pyruvate molecules. So here we see our 2 pyruvate molecules, our 3 carbon sugars. Here's another 3 carbon sugar. So there's pyruvate, and we've made the 2 NADH. So after that, it depends on if there's oxygen present or not. So where these two things go from here all depends on if there's oxygen present. So if there is oxygen present, your pyruvate and your NADH will be shuttled into the next 2 to 4 stages of cellular respiration. Now, depends on how you group them. Sometimes people put pyruvate oxidation with the Krebs cycle, so we say there's only 3 stages of cellular respiration. I like to break it into 4. So you have glycolysis 1, pyruvate oxidation, Krebs cycle, and oxidative phosphorylation. So if there is oxygen present, these will continue into these steps here. And then if there's not oxygen present, that is where fermentation is going to come in. So there's many different types of fermentation. The 2 types that we're going to look at are going to be alcohol fermentation and lactic acid fermentation. So let's go ahead and take a look and see what happens to our pyruvates and our NADH with fermentation. So when you think about fermentation, why is this happening? What's the whole point? Just like in glycolysis, the whole point was to break that glucose in half and create the 2 pyruvates. What's the point here? It all comes down to NAD+ and NADH. Now, in reality, they're really the same molecule, right? So if this was NAD+, adding a hydrogen on makes it NADH. I could break that hydrogen off. Now I have NAD+, add a hydrogen on, I have NADH. So when you look at these two, they're really one and the same. It just depends on if that hydrogen's on there, whether it's been reduced. All right, so if we have NAD+ and we add a hydrogen, we've reduced it. If we tear off that hydrogen, we're removing hydrogen, we're removing electron, we're removing energy, it becomes NAD+, we have oxidized it. So you can reduce and oxidize it back and forth here, and it'll basically, it's the same molecule though. When we look at fermentation, it comes down to NAD+ and NADH, because if you don't have any oxygen present, that NADH has nowhere to go, and it'll remain NADH, and that's a problem because they're recycling back and forth. So here's my NAD+, here's my NADH. If that NADH doesn't come back off and it stays NADH, this can't go back to glycolysis to pick up more hydrogens. So we want it to get recycled. We want that hydrogen to be used later on in cellular respiration, so that way it becomes NAD+ again, and then that can go back and go back to glycolysis and then pick up another hydrogen and keep on being recycled. So the whole point of fermentation is to remove that hydrogen at the end. So in the absence of oxygen, where the NADH can't go on to the future stages of cellular respiration, and it's just going to be kind of floating around there doing nothing, and you're just going to keep on using up your NAD+, keep on turning them to NADHs, and then eventually you run out of NAD+, that's a bad thing. That's going to stop making any energy at all. So if you look at the energy payoff phase of glycolysis here, at this step right here, this is when you make your NADH. This is when you add your hydrogen onto the NAD+. So eventually, if you didn't recycle it back, if you didn't tear off those hydrogens later on and send it back to glycolysis, you would eventually run out of NAD+. So you'd use up all the NAD+, and that would be really bad. This whole process would stop. You wouldn't be able to make any ATP at all. So really, really bad thing to happen. So our cells have a way of dealing with that. If there's no oxygen present, if you can't continue with cellular respiration, then fermentation comes in. So the first type is alcohol fermentation. Alcohol fermentation is done by your bacteria and your yeast, and there's 2 steps to converting pyruvate into ethanol, okay, which is alcohol. So you've probably heard that, you know, yeast is used in like brewing companies and stuff like that in order to make alcoholic beverages, which is true. So they actually harness the power of alcohol fermentation in order to do that. So let's take a look at these two steps. How do we go from pyruvate into ethanol? And that's what fermentation is all about. Also, though, don't forget, it's about taking that hydrogen off of the NADH too. So we want to tear off that hydrogen off the NADH and make it NAD+, and that's the whole reason why we're even doing this. So you take your pyruvate. This is highlighted in gray here because this is going to get torn off. And just by looking at it, you have a carbon and 2 oxygens. That should click something in your head right there. That's carbon dioxide. So we're going to tear off that carbon and 2 oxygens, and we're going to create carbon dioxide in the process. So we see something like this happen. Okay, we tear off this part. That leaves this part remaining. So if you take a look here, you have 2 carbons and an oxygen. We're going to add a hydrogen on here. We're going to replace this group with a hydrogen group, and we get this. Okay, so we've made carbon dioxide and an intermediate here. Well, we're not done yet because we haven't made ethanol yet. So in order to make ethanol, here's where the NADH comes in. So the NADH, which we want to recycle back into NAD+, is going to come in along with an extra hydrogen. Okay, so an extra hydrogen is going to come in as well. And what's going to happen is we're going to tear off that hydrogen, and we're going to use this hydrogen as well. When we tear off this hydrogen, it's now going to have been, again, remember oil rig, okay? It's going to lose hydrogen here, so it's losing electrons. So it's going to be oxidized into NAD+. And as a result of that, we can take this hydrogen here, and stick it on and have a hydrogen here and a hydrogen there. So if you take a look, if you study these two molecules, you have carbon, carbon, 3 hydrogens, oxygen, hydrogen. You see that we put one of these hydrogens on the oxygen here. We see that right there. And then the other hydrogen winds up on the end over here. So we have now been able to recycle NADH back to NAD+. This is going to go back to glycolysis. So that can go back and that can be used to pull energy out and continue the process of making ATP. We've now made ethanol. So it wasn't our goal to make ethanol, but that's just a byproduct of trying to recycle the NADH back into NAD+. All right, so our next thing is going to be lactic acid fermentation. Lactic acid fermentation is done by bacteria, fungus, and even us. For us, it comes down to when there's a very low supply of oxygen. If we're using up oxygen faster than we are actually getting it into the cells from our blood, then we go into lactic acid fermentation instead of continuing to the remaining steps of cellular respiration. So in this one, though, it's different from alcohol fermentation. There's only one step here. Once again, don't forget the focus is all about taking NADH and turning it back into NAD+, so that way we can go back to glycolysis and continue our process here. So here we take pyruvate. So we have our pyruvate, our 3-carbon sugar here, and we are going to add NADH and a hydrogen. So just like the other one in alcohol fermentation, so you have your NADH plus an additional hydrogen. And we're going to pull that hydrogen off, therefore oxidizing our NADH into NAD+. And then we're going to add those hydrogens on. So if you look at this molecule here, this top part of pyruvate stays the same. So if you look at here to here, nothing's changed. The bottom part is what changes. The C3 doesn't get touched, but we take one hydrogen and put it onto the end here. And we see that. We take another hydrogen, put it onto the end here, and now we've made the molecule known as lactate. So just by adding 2 hydrogens, we've changed it, and now we have lactate. Now, when you're talking about humans and lactic acid fermentation, this is going to happen during strenuous exercise. When the amount of oxygen coming in the cell, there's just not enough, and cellular respiration can't keep up, going through all the stages of cellular respiration. It used to be believed that lactic acid is going to cause muscle soreness. In fact, it was taught like that forever in biology classes. But what we recently found out is that it probably doesn't. It's probably the buildup of potassium ions that actually cause the soreness that you get from exercising, from that strenuous workout that you do. So we don't actually say that it's the lactic acid anymore. In fact, they actually found out that lactic acid probably helps during the strenuous workout, so it's a positive thing versus a negative thing and causing all that soreness. our body doesn't waste. So all this lactate that's been produced, we don't want to waste it. So when you're doing that strenuous workout and you switch over to lactic acid fermentation, all that lactate that builds up in your cells are going to get pushed out of the cells, go into your bloodstream, get transferred all the way back to your liver, and then your liver is going to convert it back into pyruvate. Well, now that you have pyruvate, you can take that pyruvate and continue on with the rest of the stages of cellular respiration. So our body doesn't waste in this case. It's going to convert it so that way it can be used in cellular respiration and get the rest of the energy out of it, because pyruvate still has a lot of energy to offer.